All right, good morning, everyone. Welcome to the first seminar for the second half of the year in the neuroprosthesis series. It's we're very lucky today to have one of our own, Dr. Jerry Silver from the Department of Neurosciences here at Case Western Reserve University. I think you'll see that there's a lot of parallels here and a lot of opportunity. So Dr. Silver does a lot of work in spinal cord injury, more in the regeneration side of things, but also in restoring respiration, which is a topic that a number of people in the FES Center have also addressed in other ways. So just let me say a few things about Dr. Silver before we get started. He has his PhD from Case Western Reserve a few years ago. Did postdoctoral work at Harvard, both in the Department of Neurosciences and in the Neuropathology Department at Harvard Medical School. He's currently a professor in the Department of Neurosciences in the School of Medicine here, has an adjunct position at the Department of Neurosurgery at the Cleveland Clinic. Dr. Silver has received a number of prestigious awards. I don't think I'm going to go through them now, but it's a big list. It's very impressive. He's on editorial boards of a lot of the top journals in the field. He does a lot of review for NIH. He does a lot of review and is on editorial boards. He's on the scientific advisory committee of the Christopher Reeve Foundation and the International Spinal Research Trust in England. And he has more than 160 publications as lead author or as senior author. So we're very fortunate to have him here today. You can see his title is Functional Regeneration Beyond the Glial Scar. So please, Jerry.
Good morning. Thanks for coming. I'm going to split the talk into two parts. And the last part is totally new. I haven't presented our work on the respiratory system ever. We've been working on this for almost 7 years. And it's finally starting to crystallize. So I'll tell you about remarkable functional recovery after long-term spinal cord injury in the respiratory system. So pretty excited about it. I'm looking for feedback. So I always start with this. Can you see my little pointer going around here? Okay, good. I always start with this slide from Ramon Cajal. He really showed us what happens after a spinal cord injury using silver stains. This is a cat. The spinal cord is cut with a knife somewhere up in here. And then he cut horizontal sections, these are the dorsal columns. And he really described what happens after spinal cord injury in terms of what the axons do. The axons die backwards or retract. Then they stop somewhere in the white matter behind the lesion. Cajal thought that the axons actually eventually die back to a sustaining collateral. But we now know that that's not true. The axons actually persist in the white matter indefinitely. And the question is why? They end in these little strange dystrophic end balls. He drew them here. They take a variety of shapes. And if the axons are entrapped in the white matter for the rest of your life, over 40 to 50 years, do these endings play some role in that entrapment process? Also, is this entrapment process also limiting sprouting? So some sprouts can occur, but it's minimal. And there's never really very much functional recovery even due to sprouting. So we would like to understand the basic biology of these phenomena. And in my talk, usually I explain the cell biology and molecular biology that surrounds these various phenomena. But I can't go, because of the respiratory stuff I want to talk about today, I can't go into that. I'll just mention what happens and summarize what we think is going on. It's a lot different than we think. So you have to open your minds, I hope. Just be open to strange possibilities. And here starts the strange possibilities. So there have been a number of theories put forth over many, many hundreds of years, actually, to explain regeneration failure. The glial scar is a mechanical wall that blocks growth. A lack of trophic substances, the water level goes down. This is Ramon Cajal's idea. There's an intrinsic lack of growth potential in the neuron. They don't have a very good growth motor. Inhibitory molecules. We'll spend a lot of time talking about that. The alligators and the sharks and the macrophages. I won't talk a lot about the macrophages. They're very important in that phenomenon of dieback. Right, that's been published now, so I'm going to have to skip that today. I'll talk about the alligators, and I'll talk about the sharks in the water. And then there's this cartoon. It's really the opposite of this one, right? Maybe there's too much of a good thing. Some weird concoction of growth factors. There's a beach chair. You might even make synapses upon it. And that I like this cartoon because it helps explain why axons can be entrapped for so many decades. But there was absolutely no evidence for it, ever. I'm going to tell you very briefly how this occurs and why this occurs. It has to do with stem cells in the core of the lesion. And so keep these in mind as we go through this. They're not mutually exclusive. All these different phenomena could happen at one time or another. The family of molecules that we've been interested in for a very long time are the proteoglycans. You may not know about them, but you've all seen them. If you go to shop at Costco, glucosamine, chondroitin, people eat this stuff, help their joints work better. It's really not true, but if you want to spend your money, that's fine. But they come in a variety of flavors. They have a protein core in the middle to which are attached these long-chain sugars called, these are the glycoaminoglycans, and there can be a lot of them, or there are a few of them, and they decorate the protein core. They're highly sulfated. They have a high negative charge. They kind of look like these caterpillars over here in scanning EM. And we first began to study them in the developing nervous system. And around the mid-1980s, I asked a really unusual question of why axons don't grow where they don't normally in the embryo. Were there such things as real boundaries or guardrails that could block the growth of axons and keep them from crossing from one area to another? That had never been asked before. We studied this area called the roof plate of the spinal cord in the embryo and the periphery of the retina. And in those boundary regions, we found the proteoglycans when this antibody called CS56 was first developed. And there in the roof plate of the spinal cord was some member of this family of proteoglycans. So the CS56 antibody sees the chondroitin sulfate sugars on whatever proteoglycan cores they decorate. And you see this is an axon stain. No axons ever cross the roof plate. We then turned to the retina where we could do some experiments and ask the question, why don't axons project out of your pupil? That would be a bad thing that can happen in certain congenital anomalies. The axons all pass through the optic nerve, which is over here, and in the periphery of the retina, this is a retinal flat mount, you can see the proteoglycans in orange, and the axons turn away from the orange proteoglycan. All right, and we knew that the proteoglycans were critical because we treated the eye cup in vitro alive with an enzyme called chondroitinase, we'll talk more about that, that clips off the sugar chains. And we already knew that the sugars were critical for the inhibition. And that really ruined the patterning in the retina. So you can see it's just abnormal. So this is in the late 80s, early 90s, and nobody believed this. These are cartilaginous molecules. What are they doing in the central nervous system? This can't be. There's no extracellular matrix in the brain. I mean, things were tough. And we published a couple of good papers in Science and Nature, but still nobody believed it. So I left development and turned to regeneration and asked the question, do these boundary molecules that we believe play a role in development reappear after injury? They downregulate during development. They can reappear in certain regions, and in particular after injury. So when we stab the spinal cord the way Monica Hall did with a knife and stain with the CS56 antibody, this is just rostral to the lesion of the dorsal columns, you see the increase in proteoglycan staining. We didn't know which one it was, but many, many years later, there are a whole family of them down in the bottom. They all appear. I'll talk a little bit about this one called NG2, but not very much today. And the whole family upregulates in the lesion. And so they're there in the right place. If you look here in this picture, cut in sagittal section, you can see the lesion in the spinal cord. And the staining in blue is the proteoglycan. And I just want to point out here that the proteoglycans distribute in a gradient. More blue here near the lesion and fading away back to background levels as you go farther away. And we know why that occurs. There's stuff that's kind of leaks out of the blood-brain barrier. It's a fibrinogen TGF-beta complex that comes out, stimulates the astrocytes and other cell types to make this stuff. And their job is to wall off the lesion. So they're building a wall, but it's in a gradient. Another place where proteoglycans are present is in the so-called perineuronal net. Now, if you've never heard of that, it was discovered by Golgi, Camillo Golgi in the 1800s, denied by Cajal, right? And then forgotten until the 1950s when specific stains became available to see these so-called ground substance. And in particular, around inhibitory interneurons, but other types, there's a dense perineuronal net around synaptic boutons throughout the nervous system. And the idea is that solidifies synapses, keeps them in place. That's a good thing, so you don't forget what you've learned each day. But in turn, the net blocks sprouting or plasticity. So proteoglycans in the scar may block regeneration, and the net may block sprouting. And we'll actually talk a lot about the net today. Okay, so I talked about the alligators and the sharks. The alligators represented in the cartoon. The alligators represented inhibitory molecules in myelin. That was a big, big story from many decades, and still is. The idea that myelin proteins block regeneration, just normal myelin. The sharks represented the proteoglycans, cartilaginous fishes. So which one is more important? I was losing this battle badly, but I thought we'd do a simple experiment, and that was to try to introduce fully adult neurons into the white matter of the spinal cord or brain very gently through a micropipette. It's called a microtransplantation. And we would just put in a few cells, a couple hundred, and kind of push them gently out into the white matter. All right? We would even make a lesion upstream to release the alligators. All right? And the idea would be that these neurons, when pushed past the scar, there's no way they could regenerate because they see all the the alligators. All right, so we did that experiment. We made a lesion in the dorsal columns. Here it is. We placed our microtransplant just rostral to the lesion among all the alligators. And surprisingly, these are adult sensory neurons from the green mouse. So they're green in a rat, no cyclosporin. And you can see the axons after several days growing like green hair. It was unbelievably robust regeneration. This is impossible. This can't happen. But it did. Right in the middle of the dorsal columns, right among all the alligators. Now, then the question was, since the axons are also growing south from the transplant site towards the lesion, what would happen when they got to the lesion itself? Would that stop them? And it does. If you lesion and then acutely transplant, or if you lesion and wait 3 full months, and actually a year after you lesion, and then place the cells into the white matter, they can still grow robustly. That was very exciting. But when the axons got to the region of the lesion, they go into the lesion and get stuck. You can see here, after 3 months, the axons now, plus 8 days. Why 8 days? This is mouse in rat. And eventually the rat figures out this is mouse, and the immune system just removes all the cells. But we've got about 8 days to work. We're 5 millimeters rostral to the lesion site, and so the axons have 5 days to get there, and they sit around, and you can see them getting stuck again. So they go into the lesion and get stuck. And the axons, which were bona fide growth cones in the white matter, when they get into the lesion, transform into these funny endings that Cajal described, looking just like this. So they transform into dystrophic shape and get stuck. So why does that happen? All right, are the proteoglycans important? So we move to tissue culture. If you look at adult sensory neurons growing on laminin in time lapse, just notice how dynamic these cells are and watch their processes going out and back. Very dynamic, growing on an extracellular matrix that they like. What happens when they see proteoglycans? And in the 1990s, we use these stripe assays. They're very simple. You just soak a piece of nitrocellulose and whatever molecules you're interested in and lay them on nitrocellulose on the culture. And then we throw down our adult sensory neurons, and in these stripe assays, axons would approach from laminin, see the proteoglycan, and then turn abruptly along the surface. Still growing very dynamically, but turning, all right? Now, this was not the dystrophic entrapped axon of Cajal, so I was never very satisfied with this kind of a model, right, in terms of duplicating the dystrophic axon, but it did allow us to test who's more important, the protein or the sugar. And so we used chondroitinase, we'll talk about that a lot later, and we saw that removing the chondroitin sulfate sugars removed all the inhibition. So we knew back in the early 1990s that the sugars were critical, but how they were critical we did not know. We thought back then it had all to do with charge, that it was a big negative charge and cells just don't like that, and that's probably true. All right, so that's not entrapment. But don't forget that proteoglycans are, yeah, question?
Is that video in real time?
Oh, no, that's sped up. that would be like a superstar.
Yeah, what's
Yeah,
the time step?
it's sped up about 250 times. But it is dramatic. Okay, remember that proteoglycans are distributed in gradients. So I thought, okay, if nature makes a gradient, we should make a gradient. Maybe the distribution is important. So I asked Veronica to make basically coffee rings. I'm a low-tech kind of a guy, and I know that if you spill coffee on linen, you get a ring. Now, people have been making coffee rings for a long time, but in 2011, finally, some physicists figured out why you get a gradient. It has to do with drying. And if you put two molecules in the coffee and cream, you get inverse gradients. All right? And so we took laminin, growth-promoting extracellular matrix molecule, and aggrecan that we can buy from Sigma, and we made coffee rings. And you can see the gradient right here. What happens is green is laminin, that decreases going outward, and proteoglycans in red with the CS56 antibody increase going outward. So there's more inhibition going out and less growth promotion. And when you coat neurons on that surface, you see the outer rim axons turn, just as I showed you earlier. But here, on the inside of the gradient, right, you get this frayed rug kind of appearance. And so the axons, you can see one in time lapse growing from this neuron right here, or a neuron like it, will actually enter the gradient just like they do in vivo when we transplant, go into the gradient and then get stuck. Now some of them bubble like this. We were very attracted to those because they're so cool and dynamic, looking like some of Cajal's pictures. And I don't have time to go into detail, but I thought, no way for 40 or 50 years axons are going to be that dynamic in the lesion. I mean, wouldn't it be nice if they were, but something else has got to go on. So we did more work. We looked for longer periods of time in vitro. And this is the work of Brad Lang. This was published in Nature in December. If you watch this film on the left, you can watch the axon growing into the gradient, and it just kind of stops and gets stuck. It doesn't bubble very much. Look at this one up in the upper left. So, there's a growth cone in the lower right. It's right here. Watch what happens. It gets resorbed and taken back retrogradely. And look at every single process. It's stuck like glue. Every one. They still move, but they can't dislocate from the dish. Look at this guy down here in the lower right. Totally stuck to the dish. Now that was really exciting because that's suggestive of entrapment. And these endings look very much like most of Cajal's drawings. And these blebs are growth cones that are taken up. Once the cell gets stuck like this, it cannot or will not produce a growth cone, right? They're all taken back up. So this was really exciting because it's entrapment. Now, this is the part of the talk I usually talk about who's the beach chair? Who is the axon stuck on in the lesion? It turns out that the beach chair are a population of stem-like cells. They're called oligodendrocyte precursor cells, OPCs, in the core of the lesion, and they make a mixture of the so-called NG2 proteoglycan, laminin, and fibronectin. And they actually make synaptic contacts upon those cells. They synapse. They make a synaptic-like connection, which is normal for those primitive glial cells to do in the brain normally. But in the lesion, it's not on a neuron, you're on the wrong cell, and they just sit there relatively happily on the NG2 cell. But I don't have time to talk about that because I want to talk about the respiratory stuff. But here's the model. So here's what happens after injury. Now, so you have to open your mind. And when you think of the word inhibition of the scar, think, I really love where I am, and that's why you're inhibited. Why leave if you've got everything you need? So after injury, you get stuff from the blood, proteoglycans are produced, and the axons enter into a dystrophic state. They are beaten backwards by macrophages, which I didn't have a chance to talk about, right? The astrocytes abandon the lesion core to form a scar around the outside. They make a barrier. And the axons die backwards until they reach the NG2 population, upon which they synapse. And that's why they sit there for the rest of your life. It's not so easy to get them off. I also want to point out that a very similar kind of a relationship occurs in the perineuronal net, where you have synapses, but now on neurons, surrounded by proteoglycans around them, kind of like a basket. Okay, so I think of this as synaptic doom. All right? Happy, but not so happy, in the wrong place. This has all been published now, so you can, if you're interested, you can read more about it. Now I want to talk about the mechanism of entrapment. Now, proteoglycans were thought to be incredibly robustly disgusting, right, because of their negative charge. But how do axons become stuck on them? Why do they grow into them? So there must be a receptor, and it has something to do with the gradient nature of what the axons see. So we needed to find a receptor, but we hadn't found one since we discovered proteoglycans in the central nervous system, in the early 90s. We had not found a receptor until I started to collaborate with this guy, John Flanagan, and his student, Angie Shen. And we found the first receptor for the inhibitory actions of proteoglycans. It's called protein tyrosine phosphatase sigma. It's a receptor phosphatase. It's part of the so-called LAR family of receptors. The LAR family was first discovered in fruit flies at the nerve muscle junction and is involved with synaptogenesis, with synapse formation. They're important synaptic receptors. The sugars are the ligand of the receptor. Interesting. Maybe some explanation of how chondroitinase actually works. So sigma is a receptor, and also it turns out LAR is a receptor itself. There's a third member called delta that we don't know too much about. So these are the first known receptor. Now, how do these receptors work? Well, the first thing we did was to look at whether the receptor does something interesting on our spot assay when the axon gets stuck. If the receptors are important for stickiness, right, this entrapped state, maybe they increase, and they do. So if you look at our spot assay and we throw down our adult sensory neurons, the cell bodies in the center are stuck. There's lots of receptor, both LAR and sigma. When the axon grows into the spot, into the rim, and gets stuck, like I showed you in the movie, then the receptor increases. It about doubles. So you can see the intense density of the receptor in the tip of the growth cone. Both sigma and LAR increase by about double. And that's quantified down here. I won't go into this. So I've already told you two behaviors that the axons do. They turn abruptly on sharp edges, right, and get stuck in gradients. And the explanation for that has to do with receptor competition. And if you're interested, you can read this paper in the Current Opinions. I wrote a paper with my son, Dan, who's over at the clinic doing a PhD in neuroscience. But he's interested in glioblastoma. So it was really great to publish with your own kid. So I want to tell you how the receptor works. So it's thought that the receptor has an extracellular and intracellular domain, spans the membrane. All the action, the phosphatase activity, is in the so-called D1 domain inside. When the receptor clusters, if it sees HSPGs, or HSPG laminin, heparan sulfate proteoglycans are a growth-promoting proteoglycan, the only one. Then the receptor becomes clustered or dimerized. And in the dimerized state, it's thought to be inactive. That is, not so stuck. And if you're not so stuck, you have more growth. Too stuck is bad. When the receptor sees CSPGs, it becomes a monomer. This has to do with the way the sulfate groups are clustered on HSPGs or CSPGs, actually. When the receptor is monomer, it's active. And active means sticky, synapse, stuck, right? And if you have too much stickiness, you're not going anywhere, all right? It's known that the stickiness is mediated somehow by this so-called wedge domain. It's right here in the D1, right? It's the wedge. It works, not exactly know how it works, but has some role in regulating the receptor dimerization. So we thought, gee, it wouldn't it be nice to switch the growth cone from this monomer state when it's stuck and fool it into thinking, right, it's a mobile growth cone on laminin or HSPGs. How do we do that? Well, if the wedge is important, let's just flood the system with more wedge. And if it sticks down here and does something, the receptor will think, so to speak, that it's dimerized. So you get the idea. It's pretty simple. We've also got to get our wedge thing through the membrane. All right, so what is our wedge thing? So we designed peptide to the wedge. Here's the wedge region in LAR, in sigma, in delta. Don't worry about delta. And so that's the wedge domain. We know that that's critical. And so we added TAT domain. TAT is based on HIV infectability and allows peptides to shuttle across the membrane. So it's a TAT peptide. And we made one to the LAR wedge, we call ILP, and we made one to the sigma wedge, we call ISP. And they're intracellular. And this is Brad, here's Jared. Jared's still here. He's at Athersys now. All my students seem to be going into industry, and they're going right down the street. So we know that the peptide can get into cells. So what does it do? So we're trying to muck about with the receptor, switch it. It works really great. So this is our spot assay. Again, we always go back to our spot assay. This is a control. You know, the axons get stuck, and I showed you that in time lapse. If we use sigma peptide ISP, at the right concentration, here's a concentration curve. At the right concentration, axons cross the rim. And that's not easy. Very few things will allow, by themselves, will allow axons to cross this very difficult bar. Because don't forget that inhibition is going up and growth promotion is going down, right? So if you have anything that allows axons to cross, it may be therapeutic because it's so difficult to get across this little rim of the spot. ISP worked and ILP worked. Now I said, I think our hypothesis was that too tight is bad. How do we test that hypothesis? If you're stuck to your chairs and there's an earthquake, right, in Cleveland, which is rare, you're going to stick to your chair. But if you're not stuck to your chair and there's an earthquake, you're going to fall out, right? So we did an earthquake assay, essentially in vitro. And so we plated our cells on a uniform substrate of proteoglycan and laminin so they can sit down and stick and grow. And if you shake the heck out of that, right, the earthquake, they won't come off because they're very stuck. But they can grow. If we add our peptide, ILP or ISP, and if it reduces adhesion, and that's a good thing, right, they should fall off when we shake them, and they do. Okay? And in the presence of the peptide, we know that they grow better because they can cross the rim of the spot. So that helps prove our hypothesis that we've actually lowered adhesion, and that's a good thing because it allows axons to grow. How does it allow axons to grow? You can see this in time lapse. So here's a typical control dystrophic ending stuck to the dish, right? It can't move, it's alive. And now we add either ISP or ILP and do time lapse, and you just watch the movie. This one in the center is kind of cool because that's the ISP peptide. Notice that the growth cone will sometimes collapse, like it did just now, but then always reform. And I told you earlier when the growth cone is stuck, right, it cannot form a growth cone. They're taken back up again, right, into those blebs. But now in the presence of the peptide, they're like tiny little, I guess, you know, kind of rocket ships. And they just never quit. And they'll reform a growth cone 30, 40, 50 times until they cross the rim. Now, wouldn't that be nice in vivo, if we can get axons to do that after injury? Now, I want to just use this. I'm not going to go into detail. This shows the specificity of binding of the peptide for the sigma receptor up here. I won't go into great detail, all the biochemistry. But I do want to point out that cells that lack the peptide, that lack the sigma receptor, right? are unaffected by the peptide, but very much affected by the proteoglycan. And these are astrocytes. Although astrocytes make their own proteoglycan matrix, they hate it because they don't have a receptor for it, and they're repelled by they follow their own backyard, so to speak, and they leave the lesion, right? And then they form a wall around the outer edge. And so astrocytes will not go into the rim at all, and they're not affected by the peptide. So proteoglycans are disgusting, probably also by their charge. But if you have a receptor to it, like Schwann cells or satellite glia, they will also crawl onto the spot, into the gradient, and they're affected by the peptide. So this gives further proof of the role of the receptor. And also, this is a spot assay using PTP sigma knockout mice. And that shows that the peptide works, all right, to get wild-type axons across the spot. Knockout cells can cross by themselves, and in addition of the peptide does not act synergistically. So this all suggests specificity of the peptide. If you're going to use the therapeutic, it'd be nice to show specificity. So now what do we do in vivo? So now we've got our animals, right, contused. We use this device to clunk the spinal cord and make a cavernous lesion. This is called an infinite horizon device. And we hit the spinal cord as hard as we possibly can with the machine. It's a severe injury to create a spinal cord kind of a cavernous lesion. It's not a complete lesion, right? It's severe, but it's not total. It's not transection. What we thought we would do is, all right, injure the animals on day 1. We'd give them a day to get to the hospital, and then we would inject our peptide. Because it has a TAT domain, we don't have to touch the spinal cord. So we would inject systemically, and we just simply do subcutaneous injections under the back skin, just above the lesion. All right, and we do that each day. We had no idea what the dose ought to be. We just picked one, right? It was relatively low. We had to start somewhere, so we picked a small amount. It's actually 11 micrograms per mil. And we went to 49 days, and then we stopped. Why? Because the animals weren't getting any better, and it was really, really disappointing. And so Brad put the animals downstairs and then kept looking at them, and they started to improve. So it takes a long, long time for improvement to start to occur. And then we started to do other kinds of behaviors. We do, it's called the BBB test. We look at walking. We're very interested in bladder function in my laboratory, so we look at urination. We did catwalk, we did grid walk, we did thermal hyperalgesia to look for pain, and then we do some physiology on the bladder. I'm going to go quickly over these observations that they've been published. What's interesting is that although the ILP peptide, the LAR receptor, was interesting in vitro, does not work in vivo, the animals stabilized at a walking score of about 10, and I'll show you a movie about what that is. But we do see an effect of ISP. See the animals in the red line start to diverge around day 49 when we stop injecting. I don't know if there's a relationship between stopping injecting and improved function, but I want to point out these are the improved animals. Notice that we have two groups. We have a group of responding animals here where the BBB score is going up. It's a 21-point scale. Some of them are almost normal, and then non-responders. So we convinced the journal that we could show our responding animals, although we can't do statistics on them. If you look at just the responding animals, which is about a third of the group, there are about 8 animals out of 26, they get to a BBB score of 16, which is unprecedented. Now, I'm going to show you how they walk. Some animals almost are completely normal. So if they respond, they don't mess around. They really get better. And I'm going to show you a movie. Here on the left is a typical control. She's between a 9 and a 10. She's got her tail down. She moves her hind legs, but she can't bear weight. And she's an 18, and she can really go. Her tail's up. She just flies across this is catwalk analysis on the bottom. Okay, so they really get better. So they can walk. They can grid walk. We looked at sensory motor coordination on a grid walk test. You can see that the feet fall through the grid. So the number of foot slips is a bad thing. And again, this is a treated animal, her tail's up, and far fewer foot slips in the ISP-treated group, and some non-responders. All right, so we always get responders and non-responders. And we looked at at the bladder. We put the animals in this cage to collect urine every single day. All right, and then it can tell us how they're urinating, and we do some physiology to look at the detrusor and the sphincter, right? They do much better. I won't go into details because I want to get to the respiratory stuff. The animals also break out into responders and non-responders. If we look at the responding animals, right, they're remarkable. So urinary frequency is way up, somewhere close to normal. As a whole, they do better than controls. But if you look at the responding animals, they do remarkably better. So responders are not, that's kind of typical of the way medicines kind of work. You get some people respond, some people don't, and we're seeing it in our animals. The question is why, what's going on? And you can ask the other question, if you respond with one behavior, if you can walk, can you also urinate or grid walk? Is there any correlation? And the answer is no, there's no correlation. If you get back one behavior, it does not mean, it doesn't predict you're going to get another one. Here's the Venn diagram. Some walk here, some grid walk, some can pee, some do 2 things, and 3 out of the 26 can do 3 things. All right? So there's a whole variety. Now, why is this happening? I'll tell you in a second. Although, if you look at all these behaviors together, and you go 2 standard deviations of a vehicle mean, 21 out of 26 animals at our 1x dose are improving in something, which is about 80%, and that's not bad. Okay, so no animals in the vehicle-treated group, achieve any recovery. How about a dose response? So we did that. So we started at this, our 1x dose. So we thought, what might happen if we give more? So we haven't gone up to 10x or 20x. It's too expensive. The peptide, we have to buy it. But we ended up to 4x. And slowly from a third x to 4x for bladder response. And so this is urinary frequency. All animals at 4x recover, 100%. And for spinal cord injured people, the ability to urinate is so important. And this is really exciting. So at a 4x dose, I have no idea what's going to happen if we go. We have not seen a dose response for walking. And I'm going to try to. So what's going on anatomically? So what we do not see regeneration from the long tracts, like the cortical spinal tract. It's not going through the injury. There's no effect on the lesion itself. There's no evidence of neuroprotection. It's the same size in both. But we do see, remarkably, is a bizarre and unprecedented sprouting of the serotonergic system below the level of the lesion. You can see serotonin in green. This is the caudal, this is rostral, this is a horizontal section, and you can see in cross-section this remarkable serotonergic sprouting in these weird blobs. So here, this is normal serotonin in the caudal cord. This is a vehicle, almost none, because there's a big lesion. These are 3 responding animals, and the pattern is completely different from section to section, right? It's like, a lava lamp, these funny shapes, and that's exactly what these look like. And also there's variability in the lesion itself, from left to right to the bottom. And so that may help explain the variability that we're seeing. This is an animal that is a non-responder. We also used a drug called methysergide. It blocks serotonin function, and you can see that behaviors that were gained decline. What we don't see is decline in the grid walk test if we use methysergide in our animals, suggesting that some other system is also sprouting and playing some role in that. We think it's the propriospinal system. And here's the pattern. It's just really bizarre. So serotonin seems to be really, really critical. We think what's going on is that there's a role of the perineuronal net. We don't see growth through the lesion. So this bizarre sprouting must mean that the perineuronal net is controlling sprouting of the serotonergic system normally, and that receptor is involved with that. So that's what we think is going on. So it's the first demonstration of the potential role of the net interacting with the sigma receptor. Okay, so how about chronic spinal cord injury? I mean that's really a challenge for the future. And the reason I'm so happy to be here, because you guys actually treat chronic spinal cord injury. But there's nothing that I know of in the biological realm that can promote regeneration or sprouting to give robust return of function at chronic stages of spinal cord injury. And I want to tell you about a remarkable new observation that involves chondroitinase in the respiratory system. So this is Pippa Warren and Warren Allalane, right, who are collaborators. Pippa's still in Cleveland. Warren's gone to Kentucky. And Warren is really interested in the respiratory system. And some of you may know this, but just briefly, the input or the efferents to the phrenic motor pool are from two major sources: the raphe nucleus, serotonin, and the rostroventral respiratory group that gets input from the Bottinger complex for pattern breathing. Those axons go downstream to the phrenic pool, either interneurons or right to the motor neurons themselves, and they drive respiration. All right? Now, if you make a lesion at C2, now you can't do this all the way across because the animals will die, so we make a hemisection lesion at C2. We cut all the axons to the phrenic pool, right, and that paralyzes, right, the ipsilateral hemidiaphragm. Now, in our model, our lesions paralyze the diaphragm forever. There are many groups, from the Gordon-Mitchell group, the Fuller group, who make smaller lesions and have spontaneous recovery. That's very important as we go forward in the talk, because our lesion is complete and they never recover, and I'll show you lots of evidence for that. So this is a complete paralysis of one side forever. Now, so what can we do to bring back respiration? Nature has put an anatomical substrate in place that we could take advantage of. It's called the cross-phrenic pathway. These collaterals that come from the opposite side, below the level of the lesion, we know are present. In 1895, Porter cut the spinal cord halfway through and then cut the phrenic nerve on the opposite side. I don't know why he did this, but of course the animal ought to die because it would asphyxiate. But because of drive, the serotonergic system goes nuts, releases lots of serotonin, and allows these collaterals that we know are there but latent to work. And this side of the diaphragm now starts to breathe. I've seen it. It happens in seconds. It's really remarkable. So we know those axons are there in small numbers and latent. So can we do something to utilize them, right? It would be nice if we didn't have to cut the phrenic nerve to get their activity. And we thought maybe if we chondroitinase treated, that would help, because we see a massive increase of the perineuronal net after injury. So nature actually increases the net. I don't know why she does that, and limits sprouting even more than it could have happened. And you can see that here with two stains of the perineuronal net. This is a lectin stain. This is a normal spinal cord. This is the CS56 antibody. You can see it around the phrenic pool. CS56 doesn't stain at all. But after a lesion at C2, there's a dramatic upregulation of some proteoglycan in the net right after injury. Away from the lesion is at C2. This increases is around the phrenic pool at C4 to 6. All right? So the net increases. So more inhibition. Well, we can get rid of that with chondroitinase by just simply injecting one shot at the level of C4 through a microneedle right after injury. And you can see that this is the area of diffusion of the enzyme and digestion of the net. It stays eliminated because the net turns over very slowly, right? It's gone for many, many months. But it comes back by about the 5th month. So somewhere between 5 weeks and 5 months, the net comes back. So it's not gone forever. But there's lots of time, for potential plasticity to occur. So we thought we would use our chondroitinase acutely and see what happens. Maybe we could restore breathing. And so these are EMG traces. This is the left hemidiaphragm. This is the right. We make a lesion. We look at the ipsilateral side. There's nothing. No activity. All right? If we chondroitinase treat, right, at day 0, of course, there's nothing. And we wait a week or 3 months. And all you get back, it's nice. It's cute. It's about 10% of normal activity. It's kind of pathetic. And we were a bit depressed about that. So we thought, all right, maybe we can bump that up. And we use this technique called intermittent hypoxia. It's great to rehab the spinal cord. For locomotion, you can use FES or treadmill training, and we all know how important that is. In the respiratory system, you simply send the animals to Denver, every 5 minutes, and then you bring them back to Cleveland so they breathe, normal air and then hypoxic air, right? And you do that for 5 minutes on, 5 minutes off, 5 minutes on, 5 minutes off, and you do it for an hour or so, right? It's not comfortable, but the animals can tolerate it, and you get this long-term facilitation, right? A lot of this has been worked out from the Mitchell lab with his collaborators. There's a clinical trial now using intermittent hypoxia in spinal cord injured people. It's really exciting. You can't do it, you don't want to do it too long, that's a bad thing. So we gave intermittent hypoxia to our acutely transected animals. Our animals do not spontaneously recover. This is post treatment in our controls, nothing. This is post saline and intermittent hypoxia. We don't see anything. But if we give IH, this is acute injury, big whopping response on the good side, and a minimal response, a couple of units that are pretty high, but not much more than chondroitinase by itself. So that's kind of disappointing. It led us to try to do other things, to build bridges around the lesion. And we had some success with that. That's published. But we put this story away. However, Warren tells me one day he found a dozen animals in the basement, in the animal room, that were lesioned almost a year and a half earlier. 12 animals with C2 hemisections. So let's just try something. So we thought, all right, we'll give chondroitinase. And in a week, we saw a little bit of recovery. We thought, oh, that's pretty cool. These animals are a year and a half after spinal cord injury. No one's ever done this because no one ever screwed up like we did. But we had 12 animals, and we thought we'd take very good care of them and use them to our advantage. And so we were very careful. So we would inject saline, wait a week, inject chondroitinase, record EMGs, or we would have a group of animals where we record and then inject the chondroitinase right away, record, and then add intermittent hypoxia or not, right? 5 animals survived of the 12. These are old animals, right? So we have 5 animals, right, to look at. It was remarkable. So if we so this is 1 week after either normoxia or intermittent hypoxia in this animal, right? These animals. This is a normoxic animal with chondroitinase. Look at the EMG activity. It looks almost normal. 3 weeks, right? Almost normal. So this is an animal that had intermittent hypoxia. Again, almost normal. And you can see the average amplitudes are almost back to normal amounts, right? It was shocking, frankly. So what's going on? We think we can see that there's an increase when we either do normoxia or intermittent hypoxia in serotonin by about double, and the net is gone. So there is an increase in serotonergic sprouting even a year and a half after spinal cord injury. So we wanted to repeat this. Is this a fluke? So we did lots more animals, but we waited 3 months. We couldn't go a year and a half, so we waited 3 months this time. Here's the baseline. Again, our animals do not recover. They're all flat. Here's the experimental protocol. We'd wait 12 weeks. We inject chondroitinase or saline. Then we start normoxia or intermittent hypoxia for 3 weeks. And it repeats again. With some animals that are, right, fantastic, so there's a big range. But nonetheless, the average amplitude is remarkable. There's a tiny little non-significant bump up using intermittent hypoxia, but we don't see any effect of intermittent hypoxia without chondroitinase in our model. But the results are remarkable. I've never seen anything like this. You can't even tell the good side from the bad side. This is the contralateral side, no effect of intermittent hypoxia. We did it again. So we wanted to see how robust this is. So this is the experimental protocol. We go 3 months. We inject chondroitinase. We wait 7 days. Then we either do 1 week, 3 weeks, or 5 weeks of intermittent hypoxia or normoxia. And again, there's a repeat of this remarkable return of function. I'm absolutely positive that this is a robust, reproducible phenomenon. It's not a mistake. It's not a fluke. We've done this 3 times now. There's a tiny little bump up of, right, average amplitude, but it's not significant from intermittent hypoxia. It's clear IH has an effect, but it's pretty small. And it has to be in the presence of chondroitinase. What's going on? Again, just the 5-HT. There's an increase in 5-HT. Right? It's measured here. So there's always this correlation of increased 5-HT and recovery of function. This is not all good news. Unfortunately, and interestingly, in a third of our animals, in the presence of IH, but not just with chondroitinase. So when we add IH, a third of the animals become completely chaotic. It's not actually totally chaotic. You can see the ipsilateral side. These are 5 examples of what can happen when we give serotonin, right? Or when serotonin, right, maybe in too much abundance, is that possible? We get what I would call chaos. It's this tonic activity. Doesn't affect the good side very much, but it can be really, really bad. These animals are not happy. They're having seizures basically in one diaphragm, all right? They don't gain weight, they don't look good. This improves over time. So this is 5 weeks now in the presence of intermittent hypoxia. They're starting to get a little bit better. There's still some inspirational, some bursting. There's spike-triggered averaging. It's not good at 3 weeks, but 5 weeks, it's a little bit better. There's still some slop in between. So they are improving over time. And in our 1.5-year animals in IH, we only saw 1 animal, right, that had some tonic activity at one point of the recording and then it went away. So it's almost as if you're paralyzed for a long time, that's better. I mean, think of the possibilities. All right, so what's going on? Again, it's 5-HT. And so what PIP did is quantify 5-HT2C receptors, 5-HT in all these sections blindly. This sticks out remarkably. I mean, in this animal, these are the tonic animals, there's a whopping amount of 5-HT, 5-fold more. So is 5-HT, is too much serotonin bad? Right? Is serotonin, a good thing and a bad thing, depending on how much it is? It makes sense that it might be bad if there's just too much. So we did this experiment. So what Pip did is to take animals, these are at 3 weeks, that are breathing fine, okay? And then she gave 5-HT systemically, and she generates tonic activity. When the 5-HT washes out, the animals recover. They breathe normally. If animals are already, they have chaos, and she gives an antagonist, I think it's called synanserin, then the chaos is removed, and she gives serotonin, the chaos comes back. So it's pretty good evidence that in too much abundance, 5-HT is a problem. And this raises all kinds of questions. I mean, is this potential development of chaos why Mother Nature inhibits regeneration? Does she know? Is she stopping things because this can possibly occur? So she stops everything, the good and the bad? I'd like to know what interneuronal subtypes are also sprouting. It can't be just this 5-HT system. There are probably some interneurons that are hooking up with the phrenic pool that don't get input from the RVRG, so they don't know anything about patterning. And when we release the matrix with our chondroitinase, maybe that is exposed, at least sometimes. Is this potential for robust recovery due to plasticity happening all over the spinal cord, in every single system, locomotor, but all inhibited by the net? Is chaos a possible alternative, right? Right, if we inject chondroitinase everywhere, or could we see improved behavior? Is it better to be chronically injured because of all this potential sprouting that's going on slowly? And can we alter the amount of sprouting using chondroitinase and our peptide and tighter things out so all animals can improve? So all kinds of cool questions. But that ends the talk with this slide from Cahal. So Cahal said, once development is complete, the sources of growth and regeneration of axons and dendrites are irretrievably lost. In the adult brain, the nerve paths are fixed, immutable, everything can die. Nothing can be regenerated. And today we heard, even after long chronic spinal cord injury, regeneration or sprouting with clinically relevant functional recovery, at least in the respiratory system, is possible. Now our challenge is to maximize this remarkable potential throughout the spinal cord. Thank you very much.
Questions for Dr. Silver?
On the first part of your talk, the stem cells are in the injured site.
Yes.
And they seem to keep these axons prisoners there, right?
Yes.
What are they doing? I mean, you would think that stem cells are there to help out.
I know you'd think so. They probably are. So the oligodendrocyte progenitor cell, the OPC, is a primitive cell that's slated to become oligodendrocytes. They sit all throughout the nervous system, in the white and the gray matter. And when an oligodendrocyte may die, they can sense that activity through the synapse that they have with the axon. So this has been discovered by Dwight Burgles, Vittorio Gallo, have studied these synaptoid structures on normal OPCs. So they sense the lack of activity and they just differentiate, make a new oligo and plug in. In the lesion core, for some strange reason, the OPCs proliferate in great abundance in the center of the spinal cord, the core of the lesion, right? And now when the axons die back, they see these cells and make a connection with them, just like they do normally. Now, why this happens, I don't know, but they love the core of the lesion. They're stimulated by inflammatory cells to grow. They don't mind the core of the lesion, and that's where the axons sit. And those cells stay undifferentiated for the rest of your life. That association just stays. I can't. It's really cool that happens, but it means that the axon is actually making a synaptic-like structure and is relatively happy for the rest of your life, and that's good. It's good news. That they're sitting in your spinal cord, if you have a spinal cord injury, forever, just waiting to be released. And also, apparently, there's a lot of sprouting going on underneath the lesion. Some of it is good, some of it is bad. That's all being inhibited. And that's happening slowly, very, very slowly. So there can be some shifting, but it's very, very slow. Again, if we release them with chondroitinase, and hopefully our peptides, so we don't have to touch the spinal cord, that activity comes out. So nature seems to want to trap axons. She wants to disallow sprouting and regeneration because of the potential of chaos. You know, people have suggested that we don't regenerate because there might be chaos. Things could get worse. And I've shown you today for the first time that actually can happen.
Well, in the brain, if you got too much regeneration, you get seizures.
Yeah, well, you could get seizures, right? But yeah, so that's inhibited. Yes.
Yeah, also in the first part of your talk, you found, well, there are 2 points. One is that you have responder and non-responder, and
Yes.
you have the sprouting of serotonin, like
Yes.
very remarkable. That serotonin, that sprouting, comes from this centrally from the nucleus, the brainstem.
Yeah. Okay. So that.
It's coming down.
No.
No, it's
No.
local. I mean, there are
So we have no evidence of growth of serotonergic fibers through the lesion and around it. Because we looked in serial sections. We do have evidence that the sprouting occurs from the remaining axons that are spared here or there around the bottom of the spinal.
are in the brain.
Well, the cells are up in the raphe, but the sprouting is all occurring locally in these weird, sharp-edged lava lamp-like patterns. We think that's occurring in that funny shape is because they're sprouting in abundance in relationship to the perineuronal net, because we haven't disturbed that with our peptide. So it's like kind of blowing up a balloon in sand, you know, and it's kind of moving its way around and then forming these weird edges. You know, serotonin pretty much is pumped out into the nervous system. I mean, it's not probably a direct synaptic kind of a thing.
It's remarkable that being just a terminal of the neurons, they can explode like that. It's amazing.
Right, they are amazing neurons.
But there is another point there. So you were relying on the mechanism of going through the blood-brain barrier, okay? And you have responded and non-responded. Did you check whether the non-responder were consequence that you didn't get into the central nervous system? For some reason, they have kinetic properties that the drug wasn't getting into. Did you try to put directly the drug into the
Right. That's obviously a possibility. There could be variability in the way the peptide accesses the central nervous system. I did show you that with a 4X treatment that bladder function in all the animals returns beautifully. So we can't, if we had more, that seems to be better. We have no idea what the upper end, we haven't done tox studies yet. We don't, with the maximum tolerated dose. There's a paper in Nature Communications in the heart. There's a heart paper. So scars, occur all over the body. And they get in the way. They block growth. In the heart model, this is after ischemia reperfusion injury of the heart. You get a scar. That drives out the sympathetic axons, okay? And that causes arrhythmia. So you can get a heart attack, you get your stent, you go home, get an arrhythmia, and drop dead, right from an arrhythmia. But in the presence of our peptide, given IP, in this model, because there's growth factors in the scar, the axons regenerate back. We made a mistake when we sent our collaborators our peptide. We forgot that they were using mouse. So we sent them the rat dose. So they gave them a dose, I have no idea how much bigger, right, than ours. It must have been 10 to 20-fold more. So if we bump up the amount, that could be very, very helpful.
I have to ask you, you sort of got my hopes raised and then dashed and then raised just a little bit about human applications. So what would it take? I mean, you're injecting these into animals. Could you inject them into people? Absolutely.
Absolutely. I don't know why it hasn't happened yet. It has to do with corporate issues, greed. Accorda Therapeutics, a company in New York, owns most of the patent rights to chondroitinase. They just haven't followed through with it. It seems to me that in a group of patients who are somewhat respiratory dependent need mechanical ventilation, maybe have some spared fibers, but not enough to get off the respirator completely. An injection of chondroitinase at C4 through a microneedle could be incredibly therapeutic. And I've just shown you the oldest reported recovery of robust function after spinal cord injury that I've ever witnessed. I mean, the amount of EMG activity is indistinguishable from the contralateral side. I mean, it seems to me that we should do this right away. We're going to substitute our peptide for the enzyme in the respiratory model, see if that might work. We have preliminary evidence that it's not so good. We have to actually combine the chondroitinase and the peptide together in chronic stages. So, but yes, I don't see why we couldn't do that. Yeah, question.
Pharmaceutical company in New York have a patent. How long has the patent been issued and does it expire?
Does the patent expire?
Company?
No, the patents, they still have patents. They have a bunch of patents on different kinds of heat-stable chondroitinases, not stable very long. There's a big effort from the spinal cord injury community to try to get this. There's a group in England, the ISRT, is funding a dog trial using chondroitinase in spontaneously spinal cord injured dogs. They get a lot of, there's a ruptured disc which presses on the cord and they get paralyzed. So there is a clinical trial, preclinical trial in dogs in England. So people are going forward despite Accorda. Yeah.
Any possibility that this may work in stroke?
Yes. We have a collaborator, Agnes Lowe, here at Case Western, who's a stroke expert, and we're supplying her our peptide for stroke. She makes cortical strokes, and we're seeing if the peptide works. Chondroitinase has already been shown to help in stroke models. The penumbra of the stroke is a potential substrate for improvement. It lacks serotonergic innervation. In the presence of the peptide, the serotonergic innervation robustly reinnervates the penumbra, and you see some improvements.
So that's
Stroke.
really subacute or acute?
Subacute.
Not in chronic. I mean,
No, we haven't done chronic stroke. This is the only work we've done in chronic spinal cord that works in the respiratory system. Wouldn't it be wonderful?
I mean, the chronic might be even more interesting.
Yeah, who knows what's going to happen? There's this issue of chaos. So we need to be careful of that. You don't want a patient, having a seizure of anything. And that's a likely possibility. So we have to learn how to control this. But if we can control it, can you imagine the possibilities in chronics? These are 1.5 year animals. You can't get older than that. The animals are dying of tumors, and this is as old as anybody's ever gone. So I hope you're as excited as I am. I think this is really cool.
Did you look at the sensory recovery in any detail? I know the BBB score will reflect that.
Well,
And I know
the grid
some of
walk, the grid walk.
Some of the responders and non-responders might be the difference between sensory recovery.
That's very possible. The grid walk test is kind of a sensory motor thing, and they do recover that, but we haven't looked at the sensory ganglia to see if they're sprouting and doing interesting things yet. But we're assuming that, and we know that the propriospinal system is doing something, because we've seen that by back labeling. So in the responding animals, we can see lots more propriospinal neurons being back labeled from below the lesion. So they're sprouting too.
Any other questions for Dr. Silver? Well, we have a little memento for you.
Oh, gee, you guys don't know how to
And
do
you know
that.
what? You're from Cleveland, but there's probably things that you don't know about. So this is what we give our speakers.
Thank you.
It's a great book, actually. So thank you.
Well, thank you. You've been a great audience.